Hello friends, so in this video we are going to study about Vacutainers. It is a very important topic from pathology and a lot of questions are asked especially in INICET examinations, and one question was asked in INICET May 2023 which I gave. Okay, the question was not about Vacutainers but the question was about order of filling of the Vacutainers. In which order will you fill it? Okay, so first of all, coming to the purple vial. In the purple vial, additive is EDTA and you must all be knowing that the use is CBC. If you don't know the uses still, no need to worry. When you will go to internship, you will easily remember all these things, that which container is used for which purpose. It will become very easy once you go to internship. Before internship, it was very hard to remember these things. Okay. Now, blue contains sodium citrate and it is used for coagulation studies. For example, PT-INR I want to check, then you will send PT-INR in a blue vial. Then ESR by Westergren's method, again blue vial. Then coming to the gray vial. The gray vial contains sodium fluoride or potassium oxalate and it is used for blood glucose level checking. Now, why sodium fluoride is added when we want to check the blood glucose level? For example, you want to check RBS or fasting blood sugar levels, then you will use gray vial because gray vials contain fluoride and fluoride is a inhibitor of the TCA cycle. That's why, and also it inhibits the glycolytic reaction in the last step. So that's why glucose which is present inside the blood will not be utilized by the glycolysis and hence glucose will be preserved in this vial. That's why we use gray vial for RBS and FBS and other types of blood sugar alcohol levels. Now coming to the red vial. Red vial has no additives. That's why blood will clot in this particular vial and we will be left with plasma, and that's why collection of serum, red vial is used. Second last is yellow containing acid citrate dextrose used for RBC preservation for HLA typing. This is important, HLA typing. Now coming to the green vial. Green vial contains heparin and is used for ABG. Of course, we all know that, and osmotic fragility testing. So this is everything that is asked from this particular topic. One more thing that is very important and is generally asked nowadays, because everyone can solve this particular part. That's why they have started asking a new type of question, order of filling. So first of all, if you are drawing blood from a patient, then in what order will you fill the Vacutainers? First of all, we will use it for culture. Okay. First of all, we will use it for culture. Why we will use it for culture at the first step? Because I don't want the needle to touch anywhere before I collect the blood for actual culturing. Because if I touch the needle in the vial and then I put the blood for culture, then it will cause false positive results. The needle may get contaminated. That's why the first blood should be given for culture. Then after culture, I will fill the blue vial, then the red vial, then the green vial, then purple vial, and lastly the black vial. Okay, black vial or gray vial. Okay, so this is the sequence. As you can see over here, this is the original sequence and you have to remember it. Now there is a way I actually made so that I don't forget it in examination, because in examination we generally tend to forget these steps and we generally confuse between two options. That's why I made a mnemonic. So what is the way? Of course, culture is the first step. So you're not going to make mistakes in that. After that, you have to remember in the name of the states. BR means Bihar. Then after Bihar, we go to Gurgaon. And lastly, we go to Punjab. Okay, PB. This B is black or gray. Okay. So this is the step. You have seen the map of India. Here is Bihar. Then here is Delhi, Gurgaon. And lastly, here is Punjab. So you start from Bihar, go to Gurgaon, and then lastly you end up in Punjab. So this is the way you can remember it in examinations.